CLO2, is an extremely powerful oxidizing agent, microbiocide, viricide, bactericide, protocide, and algicide. It disinfects through oxidation. However, unlike other oxidants, it does not produce chlorinated byproducts. Specifically, it rapidly kills microorganisms by disrupting the transport of nutrients across the cell wall. It is very important not to confuse chlorine dioxide with chlorine, as these are two distinct chemicals which react very differently and produce separate byproducts with little in common. Traditionally, chlorine dioxide is used for bleaching and paper manufacturing and has been used on a large scale to disinfect municipal drinking water since 1956. The EPA has since registered its use for food processing, including fresh produce and meat, and the sterilization of clean rooms and laboratories. Chlorine dioxide is a true gas, which means that it retains its chemical structure and properties at all times. Chlorine dioxide does not form another compound when introduced to water or other solvents. No matter what chlorine dioxide is dissolved in, it retains the same structure and reactive properties. And because it is extremely soluble in many fluids, including water and hydrocarbon, it can penetrate through material that protects microorganisms from other biocides. Chlorine dioxide is also a free radical. In other words, chlorine dioxide has an unpaired electron in its outer shell. As a result, whatever chlorine dioxide can react with, it will do so instantly, regardless of the temperature or concentration.
A complete project plan is developed and reviewed with the customer prior to the treatment date. The complete plan and timeline are reviewed, and any questions are answered at this time, and changes to the schedule or scope of work are discussed and agreed upon as well. The first step is to seal all areas to prevent gas from escaping during the treatment. This assures a safe containment of the gas as well as sufficient concentration of chlorine dioxide for disinfection. Door seams are also sealed and do not enter signage. ATP swabs and sterile swabs are taken before the treatment and once again upon completion. The ATP readings are available immediately. Launchers are placed equally around the room or area to be treated. Next, each launcher is filled with water to side gas. The units are then allowed to gas off for approximately 6 hours to assure an acceptable dwell time. Within a few hours, the areas being gassed will appear as a yellow-green color. Chlorine dioxide is very reactive and completely destroys the cell by disrupting the DNA. And when the gas has been exhausted, the units are neutralized. Within an hour, the solution is pH neutral and can be poured down the drain. At this point, the team will finalize environmental testing.